Good morning, everybody. I'm Jill Slater, and I'm going to be giving this presentation to you this morning about an introduction to international A-level biology. This is the agenda for this morning. We're going to have a look at the specification, help you ensure effective delivery of the specification. We're going to start to understand how the specification is assessed, and we're going to have an overview of the support available from Pearson. The session introduces important aspects of the course, focusing on specification and planning to teach the course. This part of the training is planned to run for 2 hours. The aims and objectives of this morning are to identify how the qualifications are devised, to review the content of the qualification, explore how to plan the course and/or your lessons, understand the assessment of the qualification and how to prepare students, to identify the support available from Pearson, and to give you the opportunity to network and share ideas with other teachers. So we're first of all going to look at how Pearson operates. Welcome to Pearson EdXL. It's the world's leading learning company and the UK's largest awarding organisation. So it's best placed to provide qualifications in line to the British educational system. The international heritage dates back over 150 years. And today, we partner with schools, universities and employers offering world-class, globally recognised qualifications to over 3.5 million students a year. We're going to start by having a look at the specification. You'll find the specification invaluable in your teaching of international A-level biology. The latest version of the specification is available to download from the Pearson website. If you look at the contents page, you can quickly locate the key information needed when planning your teaching. I would like to draw your attention to some key specification content. I'll go through this now, but the specification is available in the resources available to you, as well as downloading from the website. So the biology content is page 12 to page 42. This is the biological knowledge that you need to teach and that your students need to learn. And you need to teach all of it. The papers do not have choice questions. The aim is to cover all of the specification. The assessment information is on page 43 to 46. This section outlines the design of the assessment, including details of time and marks for each examination and details of how each unit assessment contributes to the final aggregated grade. Mathematical skills are in Appendix 6. This appendix lists the mathematical skills that can be assessed during the lifetime of this qualification. Each skill is exemplified in the appendix. However, the skills can be assessed in any appropriate biological context, and 10% of the marks across the specification go for math skills. The taxonomy is in Appendix 7. This is sometimes referred to as command words. This appendix outlines key vocabulary used in the specification, but more importantly, in the examination papers. It is important that your students are familiar with this vocabulary, and we'll look at it again in more detail later. But understanding the vocabulary helps guide them as to the type of answer that's needed for the exam question. So it's very important. if we look at the sample assessment materials, abbreviated to SAMs, these were produced when the new qualification was developed, and SAMs are examples of question papers and mark schemes, and they can be considered as templates for all subsequent external assessments used during the life of the qualification. There is a SAM for each unit examination. The SAMs can be found on the Pearson website. There are also now examples of past papers on the Pearson website, which will also give you assessment materials. I think the link's been put up for where you can download the material from. If anybody else has further problems, please let me know. If we look at how the content is assessed, the examination process can be thought of as a cyclical process. It begins with the production of a question paper. Each of the examination papers used in an external assessment is written by a principal examiner. The paper is then reviewed and revised by a committee of experienced senior examiners. So it is looked at by a number of senior examiners before it goes into final production. At the same time as the paper is produced, a mark scheme is also produced. Students then sit the examination paper and their answers are scanned into the examination software, into the marking software. All examiners contracted to mark the paper will then prepare for the exam, will then prepare for marking by looking at student answers. So all examiners will have looked at some student answers before they start final marking, and they will then provide feedback to the principal examiner as to how the questions are being answered. This feedback is then used to revise the mark scheme to take account of student responses to the questions. At this point, a final mark scheme is produced. So it may be that when the original mark scheme was produced, there were statements put in there, and then you find that students have put in other statements which are also correct, also answer the questions, and they will be added to the mark scheme. The final mark scheme is then used to standardise the marking of all the examiners. This process ensures that each examiner marks consistently. Just have a look what's in the chat box. Throughout the marking process, the accuracy of each examiner is closely monitored. This is achieved by two processes. First, a set of student responses with an agreed mark are given to examiners to mark in amongst their live marking. And they don't know when they're marking these responses that they are these monitored marking. They just marked as usual. Second, senior examiners select samples of each examiner's marking and check that the marking is of a sufficiently high standard. Once all the marking is complete, the process of awarding is undertaken. During the awarding process, candidate performance for the cohort being assessed is compared with previous cohorts and with the requirements for the award of a grade. To do this, the awarding committee balances a range of evidence. We will consider how marks are turned into grades in more detail later. Following awarding, the principal examiner for a paper will reflect on the performance of the paper. In particular, the principal examiner will look at questions that did not work as expected. Information gained from this process is then used to help refine the writing of the next paper. We'll come on to talk about the article in Unit 5 as we get later on. But it is there as part of what Edexcel put into their examination papers. If we take a closer look at the specification, to start with, we will consider the biological knowledge and understanding in the specification. We will not be looking at every specification point. There are 170 different points in the specification. Rather, we will consider how the specification is organised and how to interpret the specification. The biology content is found in pages 12 to 42. Note that the content is split into 6 units. 4 units, 1, 2, 4, and 5, detail biological knowledge and understanding that can be assessed in the examinations for these units. 2 units, 3 and 6, describe how the practical skills candidates need to develop during the course will be examined. It is envisaged that practical skills developed in Units 1 and 2 will be assessed in the Unit 3 examination, and practical skills developed in Units 1, 2, 4, and 5 will be assessed in the Unit 6 examination. So the Unit 6 examination involves all the practical skills developed throughout the whole course. Units 1, 2, 4, and 5 follow the same format in the specification. The IAS comprises Units 1, 2, and 3. The topics covered in these units are very much the foundations or building blocks required by students undertaking an A-level in biology. Pages 14 to 24 detail the biological knowledge and understanding that will be assessed in examination units for 1 and 2. If we go back, notice that each unit is split into 2 topics. The topics comprise specification points that relate to a broad theme. So, for example, in Unit 1, which is molecules, diet, health, transport, and health, the 2 topics are molecules, transport, and health, and membranes, proteins, DNA, and gene expression. Pages 25 to 27 describes the practical skills that will be assessed in the examination for Unit 3. It is important to recognise that the practicals used to develop practical skills assessed in the Unit 3 examination are described in the specification contents for Units 1 and 2. So the practicals are integrated throughout the course, and it is indicated which are core practicals, which students are expected to be familiar with the methodology for, and then there's a list of generic practical skills. Note the IA2 follows a similar pattern to the IAS. Units 4 and 5 detail biological knowledge and understanding that will be assessed in the examination for these units. Unit 6 details practical skills that will be assessed in the examination for this unit. Again, the practical skills can be developed using practicals included in the specification contents for units 1, 2, 4, and 5. So IA2 comprises of energy, environment, microbiology, and immunity. For Unit 4, and for Unit 5, respiration, internal environment, coordination, and gene expression. So the topics covered in the IA2 are more demanding than those in the IAS, bringing together further basic concepts and preparing students for the rigors of higher education. Yeah, the A level is split into IAS and IA2, and it's a modular A level. So the exams are set throughout the year, and then Unit 1, 2, and 3, the marks for those are put together to give you the IAS, and the IA2 is all 6 units amalgamated. All the marks for the 6 units is amalgamated to give the IA2. But the marks for units 1, 2, and 3 are taken for what's already been taken as an IAS exam. So that's back to the specification. Now I've gone through what's in it, but it is something you should be referring to in your planning and in your teaching on a regular basis. So we're going to look at interpretation of the specification. I'm getting questions about the IAS and IA2. The way it's constructed, it doesn't have to be done in this way, but the way it's constructed, you would expect students to teach units 1 and 2 if you're teaching it as a 2-year course, you'd expect to teach units 1 and 2 in the first year of the course, and then take the exams for those units at the end of the first year, so that you've got an IAS qualification at the end of the first year of the course, and then at the end of the second year of the course, you would take units 4, 5, and 6 and amalgamate them together to give you the IA2. The details of this are in the specification. You don't have to teach it in that way, but that is how the course is constructed. The expectation that you'd start with units 1 and 2 and the practicals for that throughout the first year, and then you'd move on to the more difficult concepts throughout the second year. There are, you don't have to do it all at the end of the year. There are exams set in October, January, and June, so you can take the exams as you finish each unit if that is the way you would prefer to do it. It's unit 1, 2, and 3 for IAS, and 4, 5, and 6 for IA2. And there is an exam for each unit. We'll look at the organisation of the exam shortly, but there is an exam set for each unit. This is in answer to questions that are put in the chat box, so I don't know if you're all looking at the questions put in the chat box, but it might be interesting to all of you. If we go back to interpreting the specification, this is an example of the first 2 pages for Unit 1. Now, you can download your own copy of the specification. Each of the units 1, 2, 4, and 5 follows the same basic plan. The unit has a short introduction. In this introduction, the philosophy or biological context of the unit is outlined. Opportunities for developing practical and mathematical skills are also identified. This introduction is followed by a brief description of the assessment for the unit. The content for the unit is then split into 2 broad topics, and each topic is split into a number of specification points. There are around 20 individual points for each topic. So it's all broken down to tell you what is expected to be covered in your teaching of this qualification. These are 2 specification points taken from topic 1. So we've got understand the course of events that leads to arteriosclerosis, endothelial dysfunction, inflammatory response, plaque formation, raised blood pressure, and then understand the blood clotting process, thromboplastin release, conversion of prothrombin to thrombin and fibrinogen to fibrin, and its role in cardiovascular disease. And then we've got, linked to that, know the benefits and risks of treatments for cardiovascular disease, antihypertensives, statins, anticoagulants, and platelet inhibitors. Both of these points relate to fairly complicated pathologies. In order to help teachers and candidates, the extent of biological knowledge and understanding for these points is included in smooth brackets. So it tells you what you need to cover. And going back, another question on the chat box. Yes, each unit has its own exam and the practical skills too. So the IAS exam comes in 3 papers and the IA2 exam comes in a further 3 papers. And as someone else has pointed out, Unit 3 is mentioned in the specification, and there's also a lab book available to you. So for Unit 3, if you're teaching an A-level biology course, you would be expected to integrate practical activities into the course. It is a science, it has practical activities. And in following these practical activities, students should be developing the practical skills that will be examined in Unit 3. We'll talk a little bit more in detail about it further later on. So if we go back to these specification points, for specification 1.11 or 1.11, candidates need to know about the role of thromboplastin, prothrombin, and fibrinogen in the clotting cascade. They do not need to be taught about regulatory factors or any details about the different mechanisms triggering the release of thromboplastin, because that is not stated in the specification. For 1.20, they need to know the benefits of antihypertensives. So they would need to know what they do, i.e. reduce blood pressure. But they would not be expected to give the name of a hypertensive or describe the mechanism of action of a hypertensive. Specification points identify what needs to be taught. Candidates will only be expected to know about the biological terms listed in the specification. The same applies to the names of enzymes, biological molecules, and biochemical details such as types of bonds. So if it's in the specification, they need to have the biological knowledge that's there. If it's not in the specification, they won't be asked specifically about those terms. However, teachers need to use their professional judgment when interpreting the specification points. This is particularly the case around biological processes. And quite often, the exam questions are set in a particular context. There will be information given about a context, and one of the requirements is students apply their knowledge to an unfamiliar situation. If there is something in the exam question that you look at and think, I've not taught that, then when you look at the question closely, you will see that the only information needed to actually answer the question is in the specification. It's putting it in a context. So if we look at this specification point from topic 6, you can see straight away, because this is topic 6, so it's part of the IA2, it is more complex. It's looking at more detailed biological information. So it says, understand the differences between the roles of B cells, B memory and B effector cells, and T cells, T helper cells, T killer cells, and T memory cells in the host's immune response. So it's one section of learning about T cells and B cells. Now I'm going to go back and ask you no, we won't. We'll go back to it. So if we look at this specification point, the idea behind it is that candidates should understand the different roles of B cells and T cells in the immune system and in an immune response. If we just consider B effector and T killer cells, it is, so we just take selecting two, the B effector cells and the T killer cells, it is reasonable to expect that candidates will be taught that plasma cells are B effector cells, and that each plasma cell clone produces antibodies that recognize a specific antigen. So if you're understanding the role of a B effector cell, then you need to know that they're produced by plasma cells and they produce antibodies that recognize a specific antigen. It would also be reasonable to teach that these antibodies are released into the blood and then bind to antigens in the blood, e.g. on the surface of bacteria or viruses, or on the surface of cells lining the blood vessels. So in order to fully explain the role of B cells in the immune system, you need to take it to the role of antibodies and the fact they bind to antigens. Equally, if we look at T killer cells, they should be taught that T killer cells have receptors that recognize specific antigens on the surface of cells, e.g. viral proteins on the surface of virus-infected cells, and that killer cells bind to the antigens on the surface of these cells and lyse them. All of that is part of understanding the role of B effector and T killer cells. However, candidates would not need to know about different antibody classes or the role of MHC antigens or the roles of other effector cells such as mast cells or natural killer cells. So if we it asks you to look up specification point 7.2. I don't know if you've all got access to the specification, so I'll put it up for you in a minute, but then I'll flick back to this, because what I'd like you to do is to think, which of these statements about glycolysis do you think your students should be taught? And which you do not think are part of the specification. So if we understand the roles of glycolysis in aerobic and anaerobic respiration, including the phosphorylation of hexoses, the production of ATP by substrate level phosphorylation, produced coenzyme, pyruvate, and lactate. Details of intermediate stages and compounds are not required. So if you could just put into the chat box what you think you should be teaching about glycolysis. Focus on what you should be teaching, and then we'll look at what you do not need to cover. I'll give you 5 minutes to actually put some ideas into the chat box. A few more ideas in the chat box. If I move it back to the specification, it gives you what I'm expecting you to think about. So just think about what you should be teaching about glycolysis. That's the specification point. I The question about newly teaching IAL Unit 4. We will look at all the support available from Pearson as we come to the end of the presentation. If that doesn't answer your question, let me know and I'll try and give you a bit more detail. Right, we've got lots of good ideas as to what you would teach. If we look at these particular statements, You would expect to teach that glycolysis starts with glucose. You would expect to teach for each molecule of glucose undergoing glycolysis, 2 molecules of pyruvate are produced. As suggested by many of you, you would expect to teach NAD is reduced in the production of pyruvate and oxidized in the conversion of pyruvate to lactate. And use the correct biological terms. Reduced and oxidized are quite important terms. You'd expect to teach that ATP is produced by substrate level phosphorylation, and that for every 2 molecules of ATP used to phosphorylate hexoses, 4 ATP are produced. You would also expect to teach the other points that you all gave, but these are things that you should be including. What you do not need to include is in glycolysis, the enzyme adding the second phosphate is called phosphofructokinase, because it says the details of intermediate stages and compounds are not required. You also do not need to teach that fructose is phosphorylated to fructose 6-phosphate. If they read and know these facts, that is useful, but there won't be marking points in an exam for these particular statements because they are not in the specification. It won't lose them any marks, but it won't gain them any credit. However, even though candidates are not expected to know something, it doesn't mean that they will never come across it. For example, given appropriate information in the examination, they could be asked to interpret some data to explain the role of a named enzyme in a biological process such as glycolysis. So in this situation, they would be applying their understanding of glycolysis and also their understanding of the way enzymes in general work. So make sure your students are aware that they will get questions where they're given information and asked to apply their knowledge in an unfamiliar context. Units 3 and 6 are organised a little differently. The specification content for Units 3 and 6 is slightly different. There's an introductory paragraph, practical skills that will be assessed and that need to be developed throughout teaching and learning. And these are listed as a set of bullet points. More details of the assessment are then provided under 3 headings: planning, implementation and measurement, and either processing results in Unit 3 or analysis in Unit 6. Within the specification are a number of core and recommended practicals. Core practicals can be considered essential in developing students' understanding of the subject and in preparing them for the practical skills assessment in Units 3 and 6. Recommended practicals are not essential, but will support student learning if they can be undertaken. Practicals should not be seen as a tick box exercise. It is important that they are used to teach students the required practical skills and enable them to perform well in Unit 3 and Unit 6 examinations. And they are also in the context of the topic that you are teaching at the time. They're not standalone activities. They are part of developing an understanding of each topic. The core practicals, the students can be asked directly about the content of those practicals, about the methodology or about the results you might expect. Whereas the recommended practicals, the questions will be linked to more generic practical skills that you expect students to develop throughout the course. Past paper practice is useful for all the papers because it helps students become familiar with how they are set out, the level of understanding they need to answer the question. But it's not a standalone. The students need to have actually carried out the practicals. As a marker, and I've been a principal examiner on the home paper, you do feel that you can tell when students have actually carried out practicals as opposed to just being taught about a practical. They may, in Unit 3, one of the questions is often to write a plan for an experiment, and it will usually be taking a core practical but modifying it based on the information that they're given in the question. So it's not just repeating a core practical, it's applying knowledge to a slightly different situation, but we will look at that in a bit more detail. So if we look at this one of the recommended additional practicals is investigate factors affecting the growth of pollen tubes. So it's an additional practical, it's not a core practical. So students, if they were asked to do anything about the methodology, they would be given some context to apply their knowledge. But this is a core practical that is very good at developing practical skills. So in the chat box, can you put what sort of factors you think might be investigated? Some good ideas there. Concentration of sugars, temperature, pH. Not so much availability of sunlight because pollen tubes grow inside the plant. So they are not going to be dependent on sunlight or light intensity. Concentration of sugar solution, very important. Wind, a bit less so because, again, they are protected inside the plant. But lots of good ideas there. In the Unit 3 examination, candidates will often be asked to devise a procedure which links back to what they will be asked about practicals. Devised procedure questions carry a relatively high mark tariff, and many students find them challenging. This type of context will usually be set in a particular context. Sorry, this type of question will usually be set in a particular context. As I was saying, they'll be given a context, asked to devise a procedure that takes their knowledge and applies it to a slightly different situation. So they will need to use both information from the question and their knowledge and understanding gained from practical work to gain the full marks. The contents of a procedure for practical is how you would carry that practical out. So if we move on and look at this is a question from the Summer 19 Unit 3 examination. The students are given some information. They're provided with a solution containing 500 grams per decimeter cubed of sucrose, and they're asked to devise a procedure to investigate the effect of different concentrations of sucrose on the rate of growth of pollen tubes using this sucrose solution. So you're given a sucrose solution and you are investigating the effect of different concentrations of sucrose. The core practical is just to investigate pollen tubes, so without changing the concentration or anything. So they've got their knowledge, or they should know how to look at pollen tubes, growth of pollen tubes, but they won't necessarily have looked at different concentrations of sucrose. So, and you're also given the results for an investigation into the effect of boric acid concentration in a sucrose solution on the growth of pollen tubes. So if you look at that, it will tell you that as you increase boric acid concentration, the pollen tube length increases up to 200 parts per million, and then it decreases. So when planning this investigation or answering this question, devising the investigation, answering the question, can you, again in the chat box, first of all, we'll do it in 2 stages. First of all, what do they need to recall from the practical skills they learned when they were undertaking practical activities in order to answer this question? So 5 minutes to put in there what they need to take from what they've learned in order to answer this question. And then we'll move on to what they need to take from the information they're given. So the practical is just investigate factors affecting the growth of pollen tubes. So they should have looked at the growth of pollen tubes. So from looking at that, can you put in a chat box what information from their knowledge and understanding they would use to answer this question? The SAMs, the sample assessment materials that are on Pearson website, as well as having questions, also have the mark scheme. For all the units, and subsequent questions on past papers, the mark schemes are also available. If you have a look at on Results Plus, that will give you a bit of information about each of the questions, and it will give at least 2 sample answers to each question with the marks that were awarded and why they were awarded. So Results Plus is a very useful tool for helping you understand what a good answer was and what a not so good answer was. And the examiner's report also has information on what was a good answer and what wasn't. Right, we're getting some examples of this. Serial dilution of sucrose solution to make 5 different concentrations. And from the practicals that you've done, you should be stating that you need at least 5 different concentrations. You'll be expected at A level to understand that anything that involves serial dilutions you should have at least 5 different concentrations. Controlling variables, yes, like type of pollen used, temperature, pH. I think the practical paper, the actual papers are available on the website, but I didn't realise there weren't any samples of answers for Unit 3. Maybe take that up with, I can't alter the exam wizard, but maybe if you put that through to your subject advisor, he will have some ideas for you. Right, if you now put into the chat box information that you would take from what's provided when you were planning this investigation, and then we'll move on and have a look at the mark scheme and what it was looking at. What nobody has put, which is sort of the basic thing for this particular practical, if you were describing how to do this practical, how you're going to measure the growth of pollen tubes, you would need to explain that you were going to use a microscope and a graticule to measure the length of the pollen tubes. So that is information that you have learned from actually carrying out this practical. And obviously you need to put in dependent and independent variables, but you actually need to state what they are. At A level, you're not expected to just keep everything the same. You are expected to say, maintain a constant temperature or maintain a constant pH. You need to actually state what you are going to change and what you're going to keep the same. Yes, volumes are important. You would want to keep the same volume, although you tend to put an excessive sucrose solution. Certain key terminology you are expected to use if you're measuring something or keeping it the same or whatever, you need to be precise. Same amount will very rarely gain you a mark. Same mass, same volume, you need to be specific. The correct apparatus, in general, you are expected to name the correct apparatus. Spelling is less important as long as it is clear what it is. The rules on spelling are that it's clear what it is and it couldn't be another biological term. So there are certain words that you've got to be really careful of, like meiosis and mitosis. You'll be expected to state that you used a graticule if you measured it, if you spelt it incorrectly, that would not be a problem. So if we move on and have a look at the mark scheme and see what. So the mark scheme was use at least 5 concentrations of sucrose solution. The sucrose is in brackets because sucrose is in the stem of the question. So you don't need to state sucrose. It just say use a range of at least 5 concentrations of solution. However, if you put something other than sucrose, that would make it incorrect. There's a mark for a detail of the dilution method of the solution that you're starting with. There's a mark for naming at least one control variable, and it has to be named. Then there's a mark for using 100 parts per million boric acid solution, because from the table that is the optimum concentration to use. There's a mark for saying that you would use a microscope and graticule to measure pollen tube length, and then stated times of measurement of pollen tube length. A sensible stated time. So it doesn't say exactly what times you've got to state, but it has to be sensible. If you said you were going to measure them every week, that would not gain the mark, because if you've done the practical, you should know that they take less time than that to grow. Now, it is, how about to give them up? It's a 5-mark question. There's 5 possible marks. They do tend to be higher tariff marks if it's a divisor procedure. You've got 6 marking points. So any 5 of those statements will gain you full marks. If you split them up into those that are from the context of the question and those that are from practical skills acquired in the course, the 5 concentrations from the 500 grams per decimeter cubed of sucrose, using boric acid at a specified concentration, and taking measurements at specified time intervals, are from the context of the question. Preparing at least 5 different concentrations, using a microscope and graticule to measure the pollen tube length, and suggesting other factors that need to be controlled, are from the practical skills acquired in the course. So the way the mark scheme is constructed means that in order to gain full marks, you need some statements from the practical skills acquired in the course and some statements from the context of the question. These questions are written so that if you just repeat exactly what you did without applying it to the context you're given, you will gain some marks, but it's not possible to gain full marks. To gain full marks, you've got to take some information from the context of the question. Now we're going to go on to have a look at the mathematical context, the mathematics content in the exams. Now, at least 10% of the marks are available for level 2 mathematics. Each exam paper, when it's written, has to have 10% of the marks available for level 2 maths. The mathematical skills that can be assessed and examples of how these skills might be assessed are given in Appendix 6 of the specification. It's very important to note that assessment of mathematical skills is not limited to the examples provided in this appendix. Individual skills shown in bold in the appendix would only be tested in the IA2 year. So they would only come up in units 4, 5, and 6. Encourage your students to be confident in the mathematical skills questions, because a lot of students panic when they see them, and they are quite capable of doing them if they stop to think about it. All of the mathematical skills can be assessed during the life of the qualification, and the mathematical skills that are not in bold can come up in any of the question papers. So if we have a look at this question, it's giving some information about the concentration of sugars in 3 pineapples. There isn't a separate section on mathematical skills. They are integrated into the questions on all of the papers. So this will follow on from a question about sugars in sugars in some context. Could be a practical context. It could be looking at possibly carbohydrates or structure of sugars. So you've got a table that gives you information from 3 pineapples, and it's looking at 3 sugars. And the question asks you to calculate the mean concentration of glucose in these 3 pineapples and to give the answer in grams per decimeter cubed. And that is in bold to help students, to send them in the right direction. Now, if we look at this question, is assessing 2 mathematical skills. The 2 skills it is testing is recognize and make use of appropriate units and find arithmetic means. Finding the mean is standard. They need to add them together and divide by the number. If they have read the question properly, they will have noticed that the concentrations are given in grams per centimetre cubed, and they are asked to give the answer in grams per decimetre cubed. It's in bold to help them make sure they don't miss it. If they don't make the conversion, I mean, there's 2 marks for this question. 1 mark will be for calculating correctly the 3 means, and the second mark will be for correctly converting it to grams per decimeter cubed. So if they miss that bit, but they calculate the means, they will get 1 mark. But this is assessing, find the arithmetic means, and also recognize and make use of appropriate units in calculations. They do need to be able to, it is a mathematical skill. Understanding standard form is one of the mathematical skills that is listed. You do need to teach them how to use standard form. If they don't, there is a risk that they will lose. It's likely to only be 1 or 2 marks across the paper, but it's still important that they know standard form. In terms of significant figures or decimal places, significant figures will usually be asked for if it's a specific number. If it's decimal places, the rule of thumb really is that your answer can't be more accurate than the information you're given to start with. So it shouldn't really be more than 1 decimal place more than the actual answer you were, the data that you were given in the first place. Encourage your students to show their working. If they get the correct right answer, they'll get full marks. No question about that. However, if it's a stage calculation, it is quite possible if they made a mistake in the later stages that they will get some marks even if they haven't got the correct answer. Or if there's a mistake earlier on and that's carried forward, sometimes there's a mark for that as well. So encourage them to show their working, but if you're marking their papers, if they've got the right answer, they can get full marks, whether they've shown the working or not. But be careful to look out for units. Percentage change is very common. Usually it will be expected to be to 1. It depends partly on the numbers, but 1 or 2 decimal places for percentage change. What I've said about decimal places, there is quite a bit of leniency when the papers are being marked. So we have sort of veered away from penalising students that have given too many decimal places in general. But in terms of accuracy, your answer can't be accurate to more decimal places than the data you are given. It would, if you get sets of data that go to 2 decimal places and you calculate the average and it comes up with 5 decimal places, that indicates that it's accurate to 5 decimal places, and it can't be, because it can't be more accurate than the information that you started with. So you should teach your students, encourage your students to give decimal places to the same number as the data they're given, or maybe 1 more than the data that they are given. If they are rounding up, which they may well be, make sure that they round it correctly. So if the number after that is greater than 5, they round up. If it's less than 5, they round down. They should, and when they're calculating percentage change, they need to be careful whether they, what's their denominator for the percentage, whether it's the change in the lower figure or the change in the higher figure. So read the question carefully, because it's surprising. We'll come on to the resources at the end, because there's a lot of resources produced by Pearson, and I think there is a math skills one, but we'll come on to that at the end, and if not, we'll follow that up. So you've got a set of data here, shows it nicely that it's accurate to 4 decimal places. It tells you that urea it tells you about urea being an enzyme. So it's not an enzyme they need to know about, but it's given the context. The reaction was described, and then the concentration of ammonia was recorded every 5 minutes for 35 minutes at a pH of 6. What level 2 math skills would you teach using this data? So very quickly in the chat box, put some ideas about what math skills you would teach. Significant figures, yes. Great. Rate of change, mean, median, mode. Yep, all sorts. So yep, appropriate units, decimal and standard form, construct and interpret tables, plot the data, calculate a rate of change from the graph. There's all sorts of things that you can teach from a set of data. And what you can do to help develop mathematical skills, you can use data sets either as a homework task or as a starter to a lesson. You don't have to, you can do it in 5-10 minute blocks or things to do at home. It's something that just should be integrated into your teaching. Now we're going to move on to think about the pre-release scientific article. I know there's been a couple of questions about that already. It is a short article of about 2,000 words. It can relate to any aspect of the qualification. It's approximately 2,000 words long. And it's accessible. The language is chosen so the language is accessible to A-level students. Although it's assessed as part of the Unit 5 examination, the content of the article and its assessment can relate to any part of the specification. The questions on the article will relate to the science that students need in order to understand the article. It's not a simple comprehension exercise. And it's only Pearson that use this method, use this type of release of an article. And it's to engender students' interest in biology as a whole. It's to give them another opportunity to apply what they know in an unfamiliar situation. And it's hopefully to increase their interest. We're now going to move on to look briefly at the command words, although we'll look at this in more detail in part 2 of the training, which is on Wednesday. It is important that your students are familiar with the command words. One of the main reasons for candidates underperforming is because they don't ask the question that's asked. And this is often because of confusion around the command words used in the question. The interpretation of command words and other issues around language are considered in more detail in the second training. However, it's worth emphasizing your students need to be familiar with these command words if they are to understand what they are being asked to do in the exam questions. Particularly things like describe and explain. If it's explain, they cannot gain full marks if they just describe. And we'll look at that in more detail next time, but there is, it is a very important aspect. Samples for the articles will be available on the SAMS sample assessment materials, and also if you look at past papers. You should be able to get those on the Pearson website. There you are. That answers some of the questions. There's a practical skills guide, a mathematical skills guide, and a topic guide. So that's an additional guide from Pearson. Now, I think you can access these slides after the end of the recording. So you will be able to go back to here and to find this link. Although I'll leave it on for just a couple of minutes so that if you want to take a picture of it with your phone, you can do that. What's in the chat box? I will put the link in the chat box at the end. Now, if we have a look at how we're going to plan to cover the course, a major concern for most biology teachers is how to cover everything in the time available. Clearly, the time you devote to teaching each particular topic or specification point will depend on many different factors, including the time you have available, the academic ability of your students, and the prior learning and attainment of your students. However, science A-level specifications are substantial documents, and the key to completing the course is to completing the teaching is course planning. These are things that you can use to help you I'll look into sending, it isn't me that would send the recorded video and links, but I will ask about you getting them sent. And thank you for somebody putting that link in the checkbox. There's several resources available on the Pearson website that you can use to help planning your teaching effectively. There's a specification, which we've already discussed, and that's a key document when planning your course. But other resources that you might find helpful are the year planner, which allows you to get an overview of how the course will be organised. The schemes of work, and there are lesson plans, all of which are available on the Pearson website, Edexcel website. You would probably start off with your year planner. And your year planner will probably include things like dates for teaching each topic. You would want to include revision dates, make sure you allow time for revision and revisiting the topic. You would want to put in, because you'd want to plan around them, the mock and external exam dates that you were using, progress review dates, You may want to put in your practical dates early on so that in case they need significant preparation. Fieldwork dates. There is an element of fieldwork which can be approached in different ways. You may take your students on a residential field trip. You may do it on off-site days spread throughout the year, and any enrichment activities that you are planning. Once you've got your year planner, you would want then to go on to plan your scheme of work. The scientific article for the next set of exams is available, and it is important that you keep on top of accessing the scientific article and plenty of time for your students. Now, Pearson produced a number of published resources, including a scheme of work. This is available on the Pearson website. This is an example of the topic of part of the scheme of work with the topic areas that are included. It's not a definitive document. You don't need to teach exactly as detailed in the scheme of work. A scheme of work is really a very personal document, and you're almost certain to want to deliver some aspects of the course differently. However, it does provide a starting point from which you can develop your own personalised scheme. The scheme of work is organised in weeks, 34 in the IAS year and 33 in the IA2. For each week, a topic area with aims and objectives is identified. Then the next column provides ideas that might help you deliver the topic. These might be examples of classroom activities or teaching resources. And then the final right-hand column identifies how the students may acquire and demonstrate transferable skills. So it gives you a starting point for what you might want to teach on a week-by-week basis. And then from that, you move on to actual specific lesson plans, which obviously depend on the time available, the length of your lesson. This particular content could be delivered in 2 or 3 lessons. If we think about what you might put in your lesson plan, here are some ideas. This identifies the topics that you're going to cover. You would identify the key concepts, the sequence of events that you're actually going to teach it, what resources are required, how you would demonstrate learning has taken place. You may also want to identify in your lesson plan the opportunities to develop math skills, practical skills, and transferable skills. However, like a scheme of work, a lesson plan is something personal to you, and it should be something that helps you deliver your teaching. Make sure it contains sufficient information to remind you what you will need to do to deliver an effective lesson. Lesson plans and schemes of work should be evolving documents. So when you get an opportunity, it is useful to reflect on how up-to-date and useful your lesson plans and schemes of work are. And it also depends on whether you have a departmental scheme of work, departmental lesson plans that are used by a number of people, how long you've been teaching A-level biology, because if you're new to it, you'll need a lot more detail in your lesson plans as you get started. And if we think about planning practical activities, many students find the acquisition of practical skills challenging. One core practical is to investigate the vitamin C content of food and drink. In the chat box, put a few ideas on the practical skills that you could teach using this practical. It's a core practical, all students are expected to do it. Very quickly, the practical skills that you might develop. You may develop practical skills. Yeah. Indicators, DC pit reaction, titration in particular. Now, you would want to carry out this practical, but you could use it to develop a different number of practical skills. You could provide a method, but ask students to devise their own investigation. You could ask them to focus on how more reliable data could be collected, such as repeating titrations and calculating mean values, or the use of error bars. You could provide alternative methods and ask students to evaluate them. Or you could give them a different context and ask them to devise an investigation based on that. Yep, good suggestions: observing colour change, tabulation, plotting a graph, planning. Or you can use this practical. It's a very good practical to use to develop a whole range of practical skills, because you can use quite precise instrumentation that should be available to most centres. There's lots of discussion about how you can improve accuracy and reliability. You can expect core practical based questions in Unit 3, but very often they will be put into a context. So this practical, all it says in the practical is investigate the vitamin C content of food and drink. Your question may be investigate the vitamin C content of a different number of a given selection of different drinks or different foods. So you apply your method, you apply your knowledge to a different situation. If we look at assessment of the course now, there are the assessment of the IAL biology is split into 6 external examinations. So the qualification, this reiterates a little bit of what I said earlier, but hopefully it clarifies it for you. The qualification can be broadly considered to comprise the AS, which would be the first year of study and comprises units 1, 2, and 3. And then the second year of study, IA2, comprises units 4, 5, and 6. All assessments are compulsory. There are no optional units, and there's no optional questions on the exam paper either. They're all compulsory. So in the next few slides, we will briefly look at the assessments for each unit. This just shows you the 6 exam papers. So for unit, The key elements of assessment for Unit 1 and 2 are shown on this slide. Both units are externally assessed. So Unit 1 is topic 1 and 2. Unit 2 is topic 3 and 4. The examination is for a total of 80 marks, and it lasts for 1 hour 30 minutes. That equates to approximately 1 minute for each mark. The topics assessed in each examination is shown in the third column. There's no synoptic assessment in these units, so they will only ask questions on that part of the specification. So, although it's not recommended, the candidates could actually take the Unit 2 examination after studying topics 3 and 4 and before studying topics 1 and 2. They are written to be sequential, but you don't have to do them in that order. Both papers have a mixture of multiple choice, short open response calculations, and extended writing questions. There are both points-based and level-based marks on the paper. There's a minimum of 8 marks, which is 10%, targeting mathematics at level 2. And there will be questions that require application of knowledge and understanding to both familiar and unfamiliar contexts. When it says it's 20% of the IAL, is this paper, each of the papers, it's got 80 marks. If you're just counting it towards the IAS, this paper counts, each of the 2 unit papers count for 40%, giving a total of 80% of the marks, and the remaining 20% is on Unit 3. If they are being amalgamated to be part of the IA2, then this particular paper counts for 20% of the marks of the IAL. I hope that explains it to you. If we look at Unit 3, this completes the IAS assessments. It's an 80-minute examination and it's worth 50 marks, which is approximately 1.5 minutes per mark. It's probably worth making your students aware of a minute per mark or 1.5 minutes per mark is not a great length of time. I'll try and help with the mark distribution again, looking at this one. So this one, so this exam is worth 50 marks, approximately 1.5 minutes per mark. It assesses the practical skills acquired studying topics 1 to 4. So there's no multiple choice or level-based questions in this exam. It's just short, open response and calculations questions. I've got a contradiction here. I've got a contradiction in what I've been told and what I think I thought there were level-based marks. There's a minimum of 5 marks targeting maths at level 2 because that's 50%. And and it requires application of knowledge and understanding to both familiar and unfamiliar questions. So it's the application and knowledge of experimental skills and experimental techniques. So if we go back to the mark distribution, you've got your IAS is a standalone qualification. But the papers taken for IAS can also be added to the papers taken for IA2 to give you an overall A-level qualification. So
哎，是不是要去接二保了？
if we're just looking at
你等会儿把下面的门关一下，我要去个厕所，会不会有点不舒服？
can you make sure you're all on mute, please? If you're looking at just the IAS qualification, it's made up of 3 units. Unit 1 and Unit 2 each count for 40% of the marks. And Unit 3, which is practical-based, counts for 20% of the marks. So the practical aspect counts for half as much as each of the other 2 unit papers. So it's 40%, 40%, and 20%. If you use your results from the IAS to amalgamate with the IA2, then it reduces the percentage of the overall qualification for each paper because you've got 6 papers instead of 3 papers. So if you're using this as a contribution to the IAL, the International A-Level, it will count for 10% of it. It's 10% of this particular paper. So the IAS counts for 50% of the IAL in total. So if we look at the Unit 4 and 5 examinations, again, Unit 4 is the same as Unit 5. Unit 4 looks at energy flow, ecosystems, and the environment. Topic 5 and Topic 6, microbiology. Unit 5 looks at Topic 7 and Topic 8. There is a mistake on this slide. So it's a written examination. It's externally assessed. Everything's externally assessed. It's a total of 90 marks and lasts for 1 hour and 45 minutes, which again is approximately 1 minute for each mark. The topics assessed are Topic 5 and 6, and then Topic 7 and 8. For both papers, between 10 and 15 marks will draw on IAS content. So there is a synoptic element to these papers. They can't forget what they've learned in IAS at the end of year 12. They've got to carry it forward. The pre-release scientific article, which is part of Unit 5, could be based on any aspect of the IAL course. So again, it's multiple choice, short open response, calculations, and extended writing question, points-based and level-based marks. Application of knowledge to familiar and unfamiliar contents, pre-release reading scientific article for Unit 5. And it's available in January, June, and October. Now, it says it's 40% of the IAS, but that is incorrect because it's not part of the IAS, but it's 20% of the IAL because that also includes the IAS. If we look at Unit 6, that completes the IA2 assessments, and it's similar to Unit 3. It's worth 50 marks, approximately 1.5 minutes per mark. It assesses the practical skills acquired throughout the course. Topics 1 to 8. And there's no multiple choice in this paper. So the IA2, all the papers in the IA2 have a synoptic aspect in them, in that they bring forward information from units 1, 2, and 3. And it's 10% of the IAL. Again, it's incorrect to put it in as being part of the IAS because it isn't. Now, if we move on to look at how grade boundaries are set, the IAL biology is awarded on a 6-point scale of A star to E, while the IAS is awarded on a 5-point scale, A to E. Each unit examination paper is marked independently. Working with raw marks, the awarding committee uses a range of evidence to set grade boundaries. And the link shows how explains a different grade setting process. But we'll go on to explain that in a minute. So each paper is regarded individually, and the grade boundaries for that paper are set. And then the grade boundaries for all the different papers are amalgamated to give the overall grade boundary. And it's the unified, the UMS, the unified marks, which are used to give the grade boundaries. So the raw mark on paper is used to calculate a final mark or UMS for the unit, and candidates are then given a grade for each unit. And the marks are aggregated. And at the awarding meetings, which we referred to at the beginning as part of the exam process, that is a meeting of all the principal examiners, the senior staff within the mark process, and the grade boundaries are then decided, bringing it in line with previous exam series. Now, we're now going to look briefly at the assessment objectives. There are 3 assessment objectives. They identify the way in which biological knowledge and understanding will be assessed. AO1, demonstrate knowledge and understanding. So AO1 questions are assessing the straightforward learning of biology detailed in the specification. If candidates were asked to explain how primary structure determines the shape of a protein, this is straightforward, out of the specification, it will be an AO1 question. The AO1 questions will account for approximately 35% of the total marks. AO2 is application of knowledge and understanding of science in familiar and unfamiliar context. So if candidates were asked to explain the role of the primary structure of the calcium channel protein in its function as a channel protein, to answer this question, candidates need to think about what calcium ion channels do, where they are found, and link the folding and location of hydrophobic and hydrophilic groups to particular places on the protein. Candidates need to apply their biological knowledge on protein structure to the specific example of calcium ion channels, so it's AO2A. And again, AO2A questions will account for approximately 35% of the total marks. AO2B is analysis and evaluation of scientific information to make judgments and reach conclusions. This includes questions to interpret data across all the examinations in the qualification. Approximately 12 to 13% of marks will be for AO2B. AO3, experimental skills in science and evaluation of data methods Any of these questions is assessing AO3. AO3 questions will be found only in the Unit 3 and Unit 6 examinations and will account for approximately 80% of the marks available across the qualification. So you can expect IAS topic questions in IAL as well. So the purpose of the mark scheme is to ensure that candidates are rewarded for correct, relevant biology and that examiners mark consistently across all candidates. The mark schemes used for each paper are published following the examination and can be a useful resource to teachers and students. Always bear in mind the mark scheme is written for a particular paper and is revised at pre-standardisation prior to its application in the marking process. So the mark schemes for AO1 questions should be reasonably consistent, but for many AO2A, AO2B and AO3 questions, candidates will need to apply their understanding to a particular context or example. And this context will change from one paper to the next, which means that candidates can't just learn a model answer. Even for questions assessing the same specification points, the mark scheme from one paper to the next may change significantly because of the different context in which the papers are set. And examiners are qualified teachers trained to mark positively and to mark as consistently as possible. So if we look at this question, describe the function of mitochondria in the movement of sperm cells. And it is a 2-mark question. The mark scheme for this is 3 marking points, which include the following points. Mitochondria carry out aerobic respiration, provide ATP or energy in order to move the flagellum. Now, the answer column gives the preferred response. That's what we're really looking for. The additional guidance column helps examiners interpret the answers candidates actually produce. So in this case, we could accept respiration rather than aerobic respiration, but we would not accept anaerobic respiration because that is wrong. And we would not accept that mitochondria or aerobic respiration produces or makes energy, because energy is not produced, it's transferred. We will accept tail instead of flagellum. We like flagellum, but we would accept tail. Now, it is useful to think about the assessment objective that is being assessed as well. In this case, the assessment objective was an application of knowledge and understanding of science in familiar and unfamiliar contexts. It's AO2A because from the specification point, they should know that mitochondria release energy and sperm have a flagellum or tail to swim, but they need to put together these bits of biological knowledge to answer this question. Now, if we think, I'll just look at the chat. They would get them if they write the answer in their own words and contain the relevant points, providing that they are using correct terminology, they would get the marks. We don't expect them to write exactly what's in the mark scheme, but we do expect them to include the relevant scientific information. And if they write a lot more than's in the mark scheme, as long as it's not scientifically incorrect, it is ignored rather than penalised. This is what the examination paper looks like. There are 6 papers for the IAM qualification. Now, this is the front cover of a Unit 1 examination paper. So the top half provides paper details and space for candidates to enter their details. Encourage your candidates to get into the habit of checking that they've been given the correct paper, as well as filling in the correct centre number and candidate number. Then it gives other relevant information about what candidates need. Then you've got the instructions, which are the same on each paper. Instructions, information, and advice. It is consistent, but it's useful for candidates to remind themselves of this information prior to beginning. Evidence from incomplete exam papers suggests that many candidates run out of time and that the majority of candidates start on question 1 and work through sequentially. So you need to think about how much time you'd advise students to spend on each question and what order you would recommend answering the questions in. And there's no right or wrong answer to this. The questions tend to increase in difficulty as you go through the paper. Then the mark tariffs also increase. It is worth advising students just to have a look at the mark tariff, but not to stop writing if they think they've written 1 mark, because they don't know exactly what the mark scheme will say. They should have enough room to give enough information to answer the question in the box provided. Now, to encourage good exam technique, students spend a lot of time revising to let themselves down with a pure exam technique. Encourage planning, especially for higher tariff questions. You don't need to fill the whole booklet. Make sure that they practice using a whole paper. Encourage students to read the question carefully. A lot of them don't. A walking, talking mock is one way to help students improve their exam technique. It's best if students sit in the same exam room where they will do their exam, preferably in the same seat. Give the student an exam paper which is as close as possible to being like the real thing, e.g. using a photocopied past paper, so they experience using the examination booklet. The most important part is to talk with students through the paper. Go through the paper question by question, explain how much time they should spend on each question. You might suggest colour coding questions according to time allocation. Draw their attention to context and suggest ways of highlighting key words. And finally, explain how there are different ways to approach an examination question. And then after you've after you've talked it through, then give them the exam under timed conditions in the way that they would normally sit an exam. After finishing the mock, you might want to provide the students with a copy of the mark scheme and get them to, and possibly an examiner's report, and get them to pre-mark each other's papers. You then might find it useful to have a discussion about some of the contentious marks that were awarded or denied. And then you could ask the students to fill out a table at the beginning and at the end to see if they feel less, the same, or more confident before and after the mark. Now, I'm going to quickly run through, and unfortunately I've overrun a little bit, so I have to do it quite quickly, the support that is available for you from Pearson. There's a number of resources available on the website, and it's worth going through them and making use of them. And the web page has 4 tabs: specification and sample assessment materials, which includes exam materials, forms and administration, teaching and learning materials, and they're constantly adding to the bank of materials, and that gives you an idea of what's available. And that shows you the website. Results Plus is a tool many teachers may find useful in analysis of their students and their own teaching performance. You can see in-depth data on the performance of a candidate, a class, or a school in reference to the cohort. You can see which questions and spec points students have performed well and poorly in, and use this in their choices around revision and future classroom teaching. It is a free online service to Pearson EdXL centres, which gives a deep insight into their exam performance. It was developed when e-pen marking technology was brought over. So students take their exam in a traditional way. Those papers are then sent to scanned in and sent to a central processing center. The electronic copies are then sent to examiners. It's not whole papers, it's question by question, which allows us to send particular questions in the same paper to be sent to different examiners depending on their expertise. They're then marked on a screen digitally so that all the data can be consolidated into a central system, which allows feedback. And Results Plus can also be used for mock exams when you're preparing for exams. Exam Wizard is another free tool that teachers might find useful in producing exercises and past papers for students. It uses questions from past papers. So the bank of potential questions increases with each series. Bespoke questions or question papers, depending on what teachers are selecting to test. There's also access to scripts on the online portal. It's a self-service, online, free, and instant from results day onwards. So it cuts down on speculative EARs, as schools have time and information needed to make informed decisions. It allows you to see all your students' marked exam papers free of charge. It's available for all qualifications and will be accessed through EdXL Online. The scripts don't have any examiner annotation on them. That doesn't come through. It's the only awarding body to offer this service free of charge. You can use it alongside Results Plus, and that gives you a detailed breakdown of your students' performance in EdXL exams. Using both these services will help you identify topics and skills where your students could benefit from further learning. Pearson Publishing also produces textbooks which support a modular approach, give exam practice. They're in line with the latest specifications, which are recognised by universities worldwide and fully comparable to UK reformed GCE A-levels. Appropriate international content puts learning in a real-world context to a global standard, making it engaging and relevant for all listeners. And it's reviewed by a language specialist to ensure the book is clear and accessible. Now, this is your dedicated student advisor. We have come to the end. You can contact him. I will just go through all the questions in the chat box quickly. I'm sorry about not being able to hear. I think somebody had not got their mute on. How to get started? I don't understand that as a question. How to get started with Results Club Plus? You can access it online. When you're signed up to teach Pearson Edexcel A-level biology, international biology, you can access things like Results Plus. You'll get a login details that will enable you to access things like Results Plus. It's obvious, it's for your students, so it's only relevant once your students have sat some exams, but you can also use it for mock exams. So you could do a mock exam and then put it into Results Plus and you would get feedback. Are there any more questions as we come to an end of this? The second session is on Wednesday morning at the same time, on the 25th. Has anybody got any further questions?